Oh, okay. No, I don't know when to start actually. Should I just start or? Yeah, in Germany too. Actually. Well, for the university lessons, not for the presentation. It's
Attivo, però stavo facendo la diretta, perché se vai sul canale direttamente sta funzionando, però non sta funzionando da quel link, proprio questo stavo vedendo. Un attimo solo. €47.
Yeah, she sent me a message.
Collegato.
Yes, hello? So, can I start now? So, welcome everybody to my talk about my PhD project. I'm Friederike Pfeiffer, and the Director of Studies is Ulisse Cardini, and my external supervisor is Professor Christian Wild at the University of Bremen. My external supervisors are Grazia Chiero and Ugo Mazzocchi. And as I said, it's a cooperation between the Stazione Zoologica and the University of Bremen. And the title of my PhD is Micropose Associations in Posidonia Meadows as Drivers of Benthic Carbon and Nitrogen Cycling in a High CO2 World. So I will now start with seagrass in general. We all know that seagrass is a very important ecosystem. And they provide habitat for many species, and they're very important for nutrient and organic matter cycling. For humans, they're also important because they provide nursery for fisheries, and they also provide coastal protection by reducing water velocity and reducing erosion. Also important nowadays with climate change, the seagrass meadows are also very important for carbon storage. So they take up the carbon dioxide and store it into the sediment. But there are also multiple threats to seagrass, which mainly humans are creating. So especially eutrophication and pollution is a big issue, but also dredging and boating from anchoring, for example, or by dredging sediments, so the water quality gets worse, and they don't have light to produce to do photosynthesis. So there are some prognosis that, for example, for Posidonia, that in 2050 there will be no more Posidonia, and actually seagrass is declining globally by 4 times the rate of tropical forest. well, these are numbers, and it's not like, it doesn't mean it's gonna be like this, but it's estimates, and for sure seagrasses are under threat as well. And so I will now, my project, as I said, it's about the high CO2 world, but I will focus on ocean acidification as one consequence of climate change. A consequence of our lifestyle, basically, of producing a lot of CO2 into the atmosphere, releasing into the atmosphere, which then also get taken up by the ocean and will lead to higher dissolved carbon dioxide. And this will again, carbon dioxide will be transferred into carbonic acid, and it will also release a lot of hydrogen ions, which then reduce the pH, and also the combination of hydrogen ion and carbonate will create more bicarbonate. And the thing is that the higher amount of hydrogen ion take up and the carbonate, which basically is used by calcifying organism, and then it's stolen basically by the calcifying organism, and to create bicarbonate. So for sure in the literature there's a consensus that ocean acidification is bad for calcifying organisms. So they are called the losers of ocean acidification, because they will they are suffering from ocean acidification. But then in the same studies they say there's also the winners of ocean acidification, which are likely the photosynthesizing organism like seagrass, cyanobacteria, and algae. But then I want to elaborate on this, because there's also some contradiction in saying those are the winners of ocean acidification. And so I was first going to tell something about the photosynthesis. So there are studies that say that seagrasses, the plant itself, can benefit from ocean acidification because there's more dissolved carbon dioxide, so they can produce more and they can do more photosynthesis. So there are some studies that found that in mesocosm, for example, that there is an increase in biomass of the seagrass, and also in the production rate and respiration, and in the increase in shoot density, for example, and the leaf growth. So it seems for the plant itself, it can be beneficial. But then if you look at the community of the seagrass, you find in several studies that the whole community is kind of degraded or impacted by ocean acidification, because it's not only the seagrass that is impacted, it's also all the organisms around it. So, for example, you have a higher rate of grazing. So even though the seagrass maybe produce more leaves, like the growth rate is higher, but then the leaf is much shorter because there's more grazing. Or we have a change in the epibion community of the seagrass leaves, for example. So they change from more calcareous organism to more fleshy algae organism. And this again can have an impact on the community and on the seagrass. So, and that's actually where my focus is lying on, is the change in the community that is living on the seagrass leaf, and how this change in the community is also affecting the change of the microbial community that is associated with it. And now I want to point out also the importance of microbial communities. So to say like why it's important to see if there's a change in the microbial community due to ocean acidification. It's because the microbial community is very important for the plant performance itself, because there are many processes that are they are done by the microbial community. And for example, the microbial film influences the uptake of dissolved inorganic nitrogen and the light availability to the plant. It also influences the nitrogen fixation, and indirectly those things will also end up in the root atmosphere environment. So it will also change the processes in the root and rhizome environment. And so my research question, how is the taxonomic and functional change of the microbial community, and how is it affected by the ocean acidification? And what role do the microbes play in the carbon and nitrogen cycle? And how is this affected by ocean acidification? And if there's any seasonal differences. So for the study side, obviously I chose Ischia because you have the naturally acidified waters because of the CO2 vents underwater in front of the castello. So this is my acidified side, and this is my control side. So at the acidified side, I find pH of 7.2, and on the control side, I find a pH of 8.1, more or less. I measured those values during 6 days for a couple of hours while I was doing my experiments. Because there's also other values in the literature which range from 7.1 to 7.6, but this is due to the high variability of pH during the day, for example. So to answer my first research question, I will look at the microbial diversity, and for this I sampled seagrass roots and rhizomes and leaves from control and acidified side, and also the water column from control and acidified side, to see how if there's a difference between the microbes in the environment and with the seagrass, associated with the seagrass. And I also took environmental parameters to see if they have an effect on this. And so I already sampled from last October. I extracted some of the DNA. Now I send it for sequencing, and I will first do 16S amplicon sequencing to see the taxonomic diversity. And then at a later step, I will do metagenomics to see what are they able to do, basically, the organism I find there. So I still need to do sequences and a bioinformatic process. And then for my second and third question, I look at the nitrogen fluxes and also at the oxygen measurements as a proxy for my carbon cycling. So I did slurry incubation. So I incubated seagrass leaves, and I looked at the processes: nitrogen fixation, denitrification, and nitrification, and so that's how my experimental design looked like. So I enriched solutions with the corresponding 50 nitrogen, and then I filled into small glass vials. I put the leaf and the solution. I incubated on a shaker, and I took in the end samples for the gas and for the nutrients to analyze. And I did this in 3 experiments. For pH, it was for acidified was 7.1. For control was 8.07 in average. So it was the same pH, more or less, that we found on our study site. So that is the experimental design, because I used the leaves I took from acidified from control. I had some which had still the epiphytes, I had some which didn't have the epiphytes, and some controls that was just the labeled water. And then I had light and dark incubation, and 4 time points, time 0 after 2 hours, after 5 hours, and after 16 hours. And so in this time I looked at the oxygen, and also I still need to analyze those samples. So I took in the end, or at the time points, I took gas samples to analyze the nitrogen gas. I took the biomass at the end of the experiments to look for isotopic ratio. I took the nutrients to look for the inorganic nutrients, and then also for the nitrification process, I took a water sample. I have to convert it into a sample that I can use to also measure the 15 nitrogen. And those are the gas chromatography and the isotopic ratio mass spectrometer, and the autoanalyzer that I use to analyze the sample. So that was another experiment. Now is the 3rd experiment, which is the in situ benthic chambers. So I measured the oxygen in situ. I created those chambers, which is basically a plastic bag and a PVC tube that you insert into the seagrass. And then inside I put the oxygen sensor to measure in situ the oxygen fluxes. Also, I took at 3 time points, I took the nutrients at the beginning, after the dark phase, and after the light phase. And at a later point, I will also try to insert label nitrogen to look at the nitrogen processes in situ. So that's how it looked like. Well, we went to the acidified and to the control side. We did the incubation first in the dark, so we put a dark cover. And then after 1-2 hours, we removed the dark cover and we had the light incubation. And then, we looked at the oxygen fluxes. And so, coming to the results of the first 3 experiments, as I said, I don't have the results yet from the microbial community, but I have some results from the ex situ slurry incubation, which is, in this case, it's the epiphyte community. You see from here, from the leaves, from the control side, you see that the leaves are very, there's a high cover of culturous algae. And then from leaves from the acidified side, the cover is not so dense and it's mostly fleshy algae growing on top of it. And then you have a significant difference in the amount of biomass in the epiphytes. So you see that there's actually a difference in the epiphytes from control and acidified side. And so I was looking, I don't have any results on the isotopic measurements yet, but these are the results from the oxygen measurements that I took at the same time. So I could look at the net primary production and the respiration of the leaves inside the incubation tubes. And I found significant difference between acidified and control, and also between plus and minus epiphytes. So, the leaves from the acidified sites produce more than the control in both cases. And also there's the importance of the epiphytes, because you can see in both cases the leaves with the epiphytes produce more. In the respiration, the difference in the control, there's no significant difference in the control between plus and minus epiphyte, but the acidified, from the leaves from the acidified, without the epiphytes, so there's still the importance of the epiphytes for respiration at the acidified side, and it's also, the respiration is lower than at the control side without the epiphytes. And then I also looked at, that was the results for the ex situ incubation. Now the results for the in situ incubation. So I was looking at the community that I find inside my chamber, and I did some measurements on the morphology of seagrass as well. So you find, I found that the leaf density, so the amount of leaves, was less on the control side, and also the amount of shoot was less on the control side than on the acidified side. However, the leaf length was much reduced on the acidified side than on the control side. So in the end, the surface area of all the leaves I could find inside the chambers was more or less the same between acidified and control. Because you have more leaves on the acidified, but you have much longer leaves on the control side. And there's it was not significant, but it seemed like there's a difference also in the epiphytes, again, dry weight. Yes. And also this picture, in the control side, you don't see, like in the acidified side, I saw on most of the shoots there was some green algae growing and some red algae and some bryozoans, and this I couldn't find in the control side. So now also the net primary production, respiration, and gross primary production of the in situ benthic chambers, I couldn't find actually any differences in this case. So, but I also had a really low number of replicates, so there needs to be more measurements done, because these are only 3 times I did the incubation, and yeah, for now I couldn't see any difference. So yeah, summing up the key results that I have so far. So the epiphyte community is different between control and acidified side, which is also confirms other studies that were done in Ischia as well. And also shoot and leaf density is higher in acidified side. It has also been shown in other studies. And the average leaf length is lower, because other studies also demonstrated that because of the grazing, the higher grazing at the acidified sites. So for the ex situ incubation, I found that the production is higher under ocean acidification. So both just for the plant, just for the leaf, and for the leaf and the epiphytes. But then I found this significant contribution of the epiphytes to the production and respiration. So I could show that the epiphytes really do have an effect, and so also the changes in the epiphytes do have an effect as well. And yeah, for the in situ incubation, as I said, I don't find any differences, and I will do more measurements in the future. These are some side projects that I was involved in the last year. One is with sea stress, involved with Jessica Fasaglia and Gabriele Procaccini. And also, this is the mesocosm experiment where they tested the effect of eutrophication and temperature rise on the seagrass. And then there's also an in situ experiment at the same time in Ischia and Bagnoli, where they test the effect of eutrophication on the seagrass. And this is with Stefanie Helber and Mirta Teichberg from Bremen University, no, from ZMT, sorry. And then I also have one side project with Phallophila, which is an invasive seagrass species, and I want to compare the microbiome from this invasive tropical species to the Posidonia species. And this is for a conference for the International Coral Reef Symposium that will happen in Bremen in July. This is my future outlook. I have to do a lot of sequencing and bioinformatics still. Then I need to do the mass spectrometry measurements for the isotopic analysis. I will repeat the in situ experiments in spring. I also will do more ex situ slurry incubations, also with the roots and rhizomes to see what's happening in the roots and rhizomes, and design a protocol how to do the labeling, to introduce the isotopic labeling into the in situ benthic chambers. And I will go to the conference of the International Coral Reef Symposium. So here I would like to thank the working group here in the Stazione with Oliso, obviously, with Luis, then my two external supervisors, Ugo and Grazia. Then I want to thank the Stazione diving team and the team in Ischia, especially Marco, Emanuele, and Antonio. And then obviously also my friends and fellow PhD students here who supported me. And yeah, of course also the people in Bremen, Christian Wild and the Marine Ecology Group over there. Okay, that's it. I put the key results for discussion at the end, and that's for now. Thank you.
Thank you. Nice study. Which are the differences between the epiphytic communities in the in the two sites?
So the main difference is the amount of calcifying algae on the leaf and the cover, because they are completely covered with this calcifying algae. And on the acidified, it's more fleshy algae, and they have less coverage, so the leaf is more open, basically.
Okay, these are the main differences.
Yeah, those are the main. I actually have to do a more close look up at all the exact biofilm, diatoms, and small algae, and yeah, I haven't done this yet, but I
but you plan to do it?
Yes.
Because I think this is an interesting aspect, to go deeper into details of these two communities.
Yeah, exactly. So yeah, I'm planning to do that as well.
Well, I think we will see also something from the sequencing, at least there, because our interest is mostly on the prokaryotic community, and those processes in the nitrogen cycle are mostly prokaryotic. So yeah, but
link to what Ulysses said. How do you prepare your samples for this, say, metagenomic analysis of the prokaryotic community? Or to say better, you are looking at the prokaryotes' presence within the Posidonia leaves, or, I mean, taking away the epiphytes?
No,
I mean, in
no.
the leaves and in the roots? I'm just curious because those are methods I'm not familiar with.
No, it's an interesting question. No, I don't look at the prokaryotic community inside, but I vortex the leaf and the roots. So I have all the microbes attached to the surface and dissolved into the water. So I take the water and I take from there I take the sequencing. So I look at the microbes attached to the roots and to the leaves.
Including the ones which are attached also to the epiphytes.
Yeah, exactly.
Yeah,
I don't
then,
scrape off the epiphytes. Okay. I put the leaves with the epiphytes, and so I look at the whole community.
Yeah, for that, a better maybe characterization of the epiphytes, I think, would be also useful. Yeah.
Yeah.
But very good, very interesting your work.
Thank you.
You told already that you worked with the mesocosm experiments of Gabriele Procaccini and his team. Now, when you do field experiments, there is always this factor that when you see a difference, then you wonder if that is caused by the factor that you are studying or some other quirks. Now, here in these mesocosms, you have better control over that. Do you think or do you plan also to do experiments under the controlled conditions in indoor mesocosms?
I was not planning to do that. I was actually focusing. I wanted to focus to measure the processes in situ more than ex situ, because I find it. I mean, the mesocosm experiments are interesting, and I think there are also studies on it already. But I wanted to see, because, as I said, there's a lot of interactions in the whole community. So I wanted to see if there's actually, if I find in situ also differences that I can really say, okay, the whole ecosystem functioning is affected by this.
I can add something, the ex situ slurry incubations that she showed, that was, the intention was exactly to do under controlled conditions. But we took the samples straight away from the field because those communities are for a long time adapted to those conditions.
That's true.
Just a curiosity. Why are the plants in the acidified sites grazed more than in the control sites? Are there any hypotheses, or they don't like the encrusting bryozoa or algae, the fishes? I don't know.
I'm not the expert in this, but I think it had to do something with phenol compound, I think, and it's more released on the acidified side, but I don't know why exactly, but that's why the grazing is higher. I think in the study of Sonino, there was explained more closely, but I don't know right now.
Other questions? The students? No? Okay. Then, Freddy, thank you very much.
Thank you. Yeah, it's true. Yeah, true. Oh, God, yeah, true. I think because I was looking at the because in the XC2 we were measuring with the micromole. I was just the XC2 we were measuring it with the micromole. Maybe eventually, yes.